Well, hello, everybody. It's Biology 101 in lockdown again. All right, we're going to go take a look at lab number 7, which is genetics. All right, how we inherit in characters. So I think it's a really interesting lab. We've got a lot of stuff going on. It's mostly a paper lab, unfortunately, but I've modified a few things, trying to make it more interesting and not as long because it's a really long lab, but we can do this. All right, what I'm going to do is switch over to the document camera so you can see the papers, and we'll start talking about what's going on. And I hit the wrong button. Buttons are close to each other. All right, here's the doc cam. Here is the lab. Come on, you focus up. Doesn't want to do it. Here it comes. There we go. Okay, so here's what the lab looks like. Again, it's on page 83, lab number 7. So you can see a lot of introduction here. Wow, they just hammered us with terms right there. So I'm going to introduce those terms in, I think, a little better way. So we're not just slamming you with vocabulary right up front. We're going to look at basic genetics, look at an allele and a gene. We'll talk about that really very soon. And then we're going to do some genetic problems, but we're going to make it fun. We're going to make it fun. That's the thing. So let me grab my notes here so I know exactly where I'm going with this. I think I would need notes, but I want to make sure that I cover everything I need to. Okay, first we're going to do a little exercise, and I'd like you to follow along. Please do that. This is really easy when this is how I introduce the topic in face-to-face lab as well. And all you need is a pair of hands. That's all you need. Pretty easy. So what we're going to do is we're going to look at a trait that you've probably never even thought much about, but that you do probably every day. And it's called hand folding. And it's just like hand folding. It's that. All right. So when you fold your hands without thinking about it, which fingers go where? All right. Where do they go? Okay. So to do this little test, you need to start off with your hands flat on a table or your lap or a chair or something. It doesn't matter where. All right. So you need to not look at your hands to start this. So what I have people do in class is we all look at the ceiling, and then I tell everybody on the count of, well, I count down from 3, 3, 2, 1, then we all fold our hands, and then you can look at your hands, but you can't look at them ahead of time, and you can't think about it. So, think of, I don't know, pizza. That's a good thing to think of. So what I do is I go 3, 2, 1, go, and everyone folds their hands without looking and without thinking about it. And then you can look at it. You can see which thumb is on top. See, my fingers are interlaced. Which thumb is on top? My right thumb is on top. Yours may be different. All right. Now what we're going to do is do it again. We're going to do it again. But this time, don't look, but think about it. We're going to try to do it the opposite way. All right. Yeah. Interlace your fingers the opposite way that you're comfortable doing it. All right. So I'm going to do 3, 2, 1, go. I go like this, and it takes longer to do it. And there it is. There's the opposite interlacing. And I have my left thumb is on top now, right? My normal way is my right thumb's on top, but my left thumb is on top. And it really took some doing without looking. It really took doing to do it. Well, what we've just done is illustrate a genetically controlled behavioral trait. And there are some behaviors that are genetically controlled. All right. So back in lab 6, I need some paper for this. Way back in lab 6, before break, we looked at chromosomes. So I need to remind you of this. All right. So we need to look at a few things because this has everything to do with this lab too. So let's get to this. Here we go. I'm just going to make a little drawing here. I'm going to draw a chromosome. All right. That is one chromosome. And you remember that I abbreviate it CHR, chromosome. That chromosome makes a duplicate of itself, and they're held together by a little belt. They're duplicates. It's still one chromosome, but there's 2 subparts now, each called a sister chromatids. And then what happens is these things twist and get shorter, just like twisting a rubber band, and they end up looking like this. right? These are the two halves here. They twist and twist and twist, and they become shorter and fatter. Well, what we're looking at in lab 7 is what's going on with these things. Well, it turns out there's a little segment. I'm just going to do this. I'm going to do this in another color. Go right there. I colored it red, this little tiny piece. That little piece is called a gene. That is a gene right there. And that gene controls has the directions for how to build a part of you. For example, the one I selected is the one responsible for making earlobes. Yep. Making earlobes. So the gene necessary to make earlobes is right there. Well, this is one chromosome that you got from one parent. Remember, you have 2 of every chromosome. Well, that means you've got a chromosome that came from the other parent. I always like to draw the female's chromosome first. I'm just going to say that came from your mom. This one came from your dad. Here we go. And the same chromosome, but from a different parent, right? They're both part of that same chromosome pair. Well, that chromosome also has a little segment on it that controls the trait for earlobes. That is our gene, right? For earlobes is in the same place. Together, these make up a homologous pair. They're the same chromosome, but they came from different parents. All right, pair, singular, right? This is a homologous pair. So from mom, from dad, and that makes up your homologous pair. All right, so you have 2 copies of these chromosomes. Now, your earlobes can be quite different, right? From if you look around at people, some people have an earlobe that, let me go ahead and make a little sketch here, an earlobe that looks like this. I'm gonna make, here's their ear, and they've got a big old lobe hanging down, all right? And we call that an unattached earlobe or free. I'm going to call that free. So it's either called free or unattached. All right, so they dangle down. All right, now other people have an earlobe that goes like this. Just kind of goes down the side of their head instead of looping up and forming an earlobe. These are called attached earlobes. And yeah, they can have a little bit more of an earlobe than what I drew, maybe a little bit here, but it goes down. You don't have this looping back up. That's an attached earlobe. All right, well, it turns out that you get one copy of the earlobe gene from your mom and one from your dad. They don't have to be the same one. They can be different, right? You can get the unattached or free one from mom and the gene for attached from dad. Those 2 different versions of the same thing are called alleles. These are alleles. So this is the gene for earlobes, but the condition or the allele of this one is free, also called unattached, where if you inherited this one from your parents, then you have the alleles for attached. All right, hope that makes kind of some sense. What I'm going to do is make a little table because I really like making tables. They're very good. So we're going to go like this. We're going to go, I think I'll change my paper as well. So hang with. Here we go. We're going to do, here's top of my paper here, right over there. Okay. We're going to go that you got, here's mom. All right. We're going to do mom. So you got from mom the trait for free earlobe. All right. Let's say that from dad, you also got the trait for free earlobe, all right? This means that you are a combination of mom and dad. You just go plus. Mom plus dad equals you. So we're going to go, all right, what are the traits here? Let's write down, I'm going to write down you, okay, you. And so you have free plus free equals free. Okay, free earlobe. Well, if you inherited the free trait from mom and the attached trait from dad, it turns out that what you have there is a free plus an attached. And what that equals is free. Seems kind of interesting, huh? Even though you inherited attached from dad, it doesn't matter. It's not going to show up in you. You're going to have free earlobes. But what if you inherited the attached from mom, the attached from dad, then you are attached and attached. Well, that equals attached. So clearly something's going on with the relationship between these. All right. So what's happening here is what you look like is what we've described here. Excuse me. All right. This is the appearance, your appearance or the expression of that gene, I should say of that allele, that version of that gene. All right. These are the expressions, and we call those the phenotype. Phenotype. Okay. And the way I tell people to remember it, it's really simple. Phenotype is your face, right? What do you look like? That's your phenotype. Yeah, it's your expression inside as well as outside, but phenotype, face, that's a great way to remember it. It really is. Now we need to look in here. We use letters of the alphabet to designate which of these we're talking about. It's all for communication. There's not a big letter stamped on your chromosomes. So here, the trait for free earlobe is this. It's going to be a capital U, unattached. Okay. So here's U. So this really would be U plus U, or we just draw it UU. We don't do the plus sign there. And then you are going to be this. You're going to be that two copies of the same trait. All right. So here's another U. Here's attached. Now, here's the thing. Notice that this one here, the attached, doesn't actually show up in the person. I'm going to lower this light a little bit. It's a little bright. And so maybe I'm going the wrong way. Yeah, it is. This thing's upside down. Okay, so when these are together, attached doesn't show up. That means that free dominates attached. It dominates it. Take this down one more. There we go. So this one's said to be the dominant allele. This one's said to be the weaker or dominated allele. Better yet, we call it the recessive allele. And we illustrate it by using a lowercase character like this. So you, if you have a free allele and an attached allele, you would be this, capital U, lowercase u. And notice that the two traits are different. These ones are the same. These ones are different. Let's look at this one here. Now here is that recessive trait, right? And we're going to designate it by using a lowercase u and a lowercase u for that one. This means that you would be lowercase u attached. All right. So here we have two of the same. Here we have two of the same. Here we have the two are different. And the terms we use for these are incredibly important. But before I introduce those, let me talk about these letters. These letters are representing the actual condition on the chromosome. And because it's the gene, which is part of your chromosome, we call these your genotype. Genotype, all right? Starts with a G. So your genotype would be this right here. It would be capital U, that's your genotype. Or if you're this one, you'd be this one. Or if you were this, you would be that genotype. These three things are genotypes. Now, to designate these, we of course have $10 words. We're going to need to know those words because we use them all the time in genetics. All the time. I'm sure you've heard them before. This one, because they're the same, well, what's the prefix for same? It's homo. We're talking about chromosomes and talking about you, the result of being a zygote. We say that this is the homozygous trait, homozygous, but it's dominant. So we say it's homozygous dominant. It'll make more sense in just a minute. This one, because they're different, well, the prefix for different is hetero. So this is heterozygous. We don't need to qualify it. This one here is also homozygous. They're the same, but they're recessive. So we say recessive. Okay, you need to internalize those terms. Homozygous dominant, well, that's our sloppy M, huh? Heterozygous and homozygous recessive. All right, don't fall into the trap of thinking you need to modify the word heterozygous. I have heard students say heterozygous dominant. No, just heterozygous, that's all you need. Okay, these are the conditions that we're looking at. Now remember, these are parts on a chromosome. All right. Okay, so where am I at here? All right, earlobes. Now, earlobes are a case of something called complete dominance. Complete dominance, because that free trait is completely dominant over that recessive trait. So if somebody is heterozygous, that homozygous dominant allele is completely dominant over this. You don't even know if someone is this. And in fact, if you look at somebody, you don't know if they're heterozygous or if they are homozygous dominant. You can't tell. Okay? In this system, in complete dominant system, you can't tell if someone is one or which one of those they are. The only way to tell in this complete dominant system is for them to have offspring and get a certain result. All right. If this recessive trait appears in any of their offspring, then you automatically know they're heterozygous. Otherwise, you just don't know. Okay. Now, in some traits, these combine, and this is where it gets kind of fun because they can combine this way. I'm just kind of getting ahead of the whole thing, but this is just too fun not to say it now. So here we look at hair curliness. It's a really fun one to look at. So here's a person here, and I'm not good at drawing people. I can draw insects and plants just great. People, nope. So here's a person that has really curly hair, all right? Person with really curly hair. I'm just going to say curly, and I'm going to say that curly is dominant over other forms. And I know that this person that has curly hair is homozygous dominant, and I'll tell you why. Because people that have straight hair, all right, people that have straight hair, turns out, are homozygous recessive. And in this system, not complete dominance, but in this system, the person that is heterozygous, that is this here, heterozygous, so they have one of each, these things actually blend together, and this person has what's called wavy hair. Wavy hair. And waviness comes in many, many degrees. All right. If your hair is completely straight and you know who you are, you curl it and nothing you do can make that curl stay unless you use some pretty nasty chemicals that aren't good for you. Your hair is perfectly straight. All right. If you have really curly hair, you're not going to be able to straighten it. All right. I don't know why some people just aren't satisfied. I once had a friend who had beautiful, long, black, straight hair, absolutely beautiful hair. And we were going, 4 of us back in high school, went to Magic Mountain, literally when it first opened. Yep. And so we go to her house to pick her up, and she's just not coming out. You know, we're all waiting in the living room, and turns out she was straightening her hair. She'd spent like an hour straightening her hair to go to Magic Mountain. It was hilarious. Okay, great. So we get up there and we get into the log ride, and you guys have been on the log ride before, right? And as you go up and you're going in the log ride, you go in this little funny house, a log ride. And as we're going through there, she starts screaming, ah, my hair, because what they do in the log ride is they mist you just before you go down the hill. So we're going through this misting tunnel, and we come out the other end and her hair's all straight again. All right. You can't change genetics. You just can't. So this is a system where they blend, and we're going to talk about that stuff later. I just thought I'd get ahead of you and try to generate a little interest because this is a lot of fun. Lots of people have wavy hair. I have wavy hair. Always wish I had straight hair because wavy hair sometimes is pretty unruly. All right. Anyway, those are some of the things we are going to take a look at today. So moving on here. Okay, now it's going to get kind of messy. But you know what I'm going to do instead of hitting up some more notes is that I'm going to show you some real live examples so that we can have some fun with this. I don't know if you ever watched this show This guy, Eugene Levy, he's a great actor. He's absolutely hilarious. He's been in so many movies. This is his real life wife in the show called Schitt's Creek, not the word you think it is, spelled differently, which was just on for a few years, just ended last year. He and his son, Dan, wrote it. I mean, look at these guys. Are they amazing? He is a carbon copy. Dan's a carbon copy of his dad. Also, Dan's sister, Sarah, is in the show as well. She doesn't play his son. Dan plays his son in the TV show. Sarah plays a waitress in the show. This is Eugene's real life wife, so she is the mother of these two. All right, so we look at interesting traits, and what Eugene's really known for are those bushy eyebrows. That is not makeup. That is a real life genetic condition that some of you listening may have. And it's having really, really, really bushy eyebrows. See, look at Dan right there. But look at Deborah, thin or called fine eyebrows, and Sarah, fine eyebrows as well. All right. Well, what's going on here? All right. Well, it turns out that to have fine eyebrows, which is recessive. Let me try to refocus this thing, guys. Hang on a sec. It's not your eyes. My little doc cam, it's still a little imperfect, but whatever. So she's got this homozygous recessive condition. So she must have received from her parents, from each parent, that recessive condition. That's the only way you can have fine eyebrows. All right. Now, bushy eyebrows are either homozygous dominant or heterozygous. Remember, I said you can't tell someone apart when they have these. The only way you could tell is if they have offspring. And if the offspring actually have the recessive condition, then you know they were heterozygous. Well, take a look. Here is Dan, who clearly has bushy eyebrows. So his genotype has to be homozygous dominant or heterozygous. And here is Sarah, who has fine eyebrows like mom. She must be homozygous recessive. All right. Let's go back to the parents. Okay. The only way Sarah could get 1B and 1B, well, first off, she got that one from her mom, clearly. You get 2 copies of every trait. Well, where'd that one come from? It has to be dad. Therefore, dad is heterozygous. There we go. All right. Well, this is fun too, because Dan, because he's also bushy, he's either homozygous dominant or heterozygous, but we can figure it out because now we know the dad's genotype. So the dad gave Dan the B trait, capital B, the dominant trait came from dad. I'm gonna write dad next to that. Well, what did his mom give him? Well, his mom only carries the trait for fine eyebrows. So what did Dan get from his mom? He got the recessive trait. Dan, therefore, is also heterozygous. Isn't that fun? You can figure out simple stuff like this just by looking. We didn't have to do anything other than know how the system works and take a look at the people involved, and you can get it. And I thought it was pretty fun. I put this thing together. It's also on Canvas, so if you want to check it out and think about it yourself, there it is. Okay. Oh, we're not done though. There's more stuff. There's more. All right. Let's look at eye color before we really dive into it. I also have a keynote presentation I want to show you that's part of lab I put together that's strangely not in the lab itself. So I threw something together I think is really nice. All right. Let's look at eye color because this is pretty interesting stuff too. Here is mom, who has brown eyes, which we're going to register that as not blue. And here is also a dad who has brown eyes that are not blue. All right. You'll understand what I mean in just a minute. Either your eyes are blue or they're not blue. Doesn't matter what color they are. I've had students go, what about green? Green is not blue. So either blue or not blue. That's the trait. That is one of the genes. All right. So they have kids, and we have here that the kids have brown eyes. This one kid has brown eyes. This one has brown eyes, not blue. All right. There's no blue trait here. So the genotype for mom must either be homozygous dominant or heterozygous. Same for dad. Same for both kids. Look at that. If I scroll down here, you can see, I'm going to ask you, what are the genotypes for each person? Well, I did put it up here just so we could do this quickly. And then I listed them here. And that's what you're going to need to do on the lab. And I'll go over that later, is list things like this. And then notice that when they have the dominant phenotype, when they have brown eyes or just not blue eyes, we can't determine their genotypes. We can't. All right. Can these children be the offspring of these parents? Yes. Yes, they can, because this person could get both capital E's from their parents, or they could get a capital E and a small e if one of the parents is heterozygous. All right. These could absolutely be the children of these people. All right. This is a little box, a little multiplication table invented by a scientist whose last name was Punnett. Invented it in about 1925, if I remember correctly. And what you do to figure out what kind of offspring two people or other living things can have is that you fill out the multiplication table. Now, here we have a mom. Now, if we assume that both parents, we're going to assume both parents are homozygous dominant right here. Well, that means that mom, I'm going to write mom across the top and dad over here. That's how I always do it. I always do the female across the top and the male in this column over here. That's just my standard, doesn't matter. So if here's mom, and let's assume that mom is homozygous dominant. Well, that means when she goes to make ovules, reproductive cells, she's going to start off with her stem cell here, which is going to eventually split in half, right, like this, and make more cells. And what's going to happen here is that 50% of the reproductive cells she makes carries that non-blue-eyed trait. Well, the other 50% also carries the non-blue-eyed trait. I know you're going to go, why don't you just write 100%? Trust me, if we go 50-50, the genetics problems go so much better because you won't make a mistake. So when you do these tables, you have to split it into 50-50 and put them here. So 50% of the ovules mom makes carry that homozygous dominant trait right there, that I should say that dominant trait, capital letter E. Well, the other 50% carries the same, right? Dad, we're going to assume dad is also that way. So 50% of his sperm cells are carry the trait for non-blue eyes, and the other 50% also carries the trait for non-blue eyes. So what we do is we write a capital E here, and just to make it really obvious what we're talking about, I like to draw, that should be an oval, I like to draw a little tail going, that's an ovule, that's a sperm cell, because that's what we're talking about. Makes it so much easier to do these when you just draw it in. Ovules, sperm cells, there we go. So what happens if this sperm cell fertilizes that ovule? Well, what is the genetic combination? Now, we're not going to draw the cells anymore in here. We're just going to draw the genetic results. We're going to draw that. There's the resulting thing. That's the new cell. I could just draw a cell and say that's a zygote. There we go. That works, but it's going to make the page messy. Okay, what if a sperm cell carrying that trait fertilizes an ovule carrying that? It's going to be that. What if a sperm cell carrying that trait fertilizes an ovule carrying that trait? Well, I know it's going to be the same. What if a sperm cell carrying this trait fertilizes an ovule carrying that trait? It's going to be that. Oh, look at that. So now we have 100% homozygous dominant. So what's the chance of them having a kid with blue eyes? 0. What's the chance of them having a kid with non-blue eyes? 100%. Now here we can assume that both parents are heterozygous. All right, what if they're heterozygous? So here's the ovules, here's the sperm cells. That's better, make it a little oval. There we go. So what if this sperm cell fertilizes that ovule there? You get homozygous dominant, that's another zygote. What if a sperm cell carrying the dominant trait fertilizes an ovule carrying the recessive trait? It's going to be this. Oh, that's interesting. Heterozygous. What if a sperm cell carrying the blue-eyed trait fertilizes an ovule carrying the non-blue-eyed trait? It's going to be the same. We always put the dominant letter first. Doesn't matter which one's which. We just put the dominant one first when it's heterozygous. Okay. What if a sperm cell carrying the trait for blue eyes fertilizes an ovule carrying the trait for blue eyes? It's going to be this. There we go. All right, how many possibilities? 1, 2, 3, 4. So we're going to put that on the bottom of a fraction. We're going to say 1 out of 4, well, not 1. We're going to count how many of these have blue eyes. Nope, nope, nope. Yep. 1 out of 4 blue eyes. The chance of this couple, if this is their genotypes, both of them, the chance of them having a blue-eyed child is 1/4 or 25%. They have a 25% chance of having blue-eyed child. What's the chance of them having a non-blue-eyed? Would be 1, 2, 3 out of 4 equals 75% chance of having a non-blue-eyed child. Okay, so we can figure this stuff out. And now we can actually effectively answer this bottom one here down there. Can these children be the offspring of these parents? Sure, yeah, 1, 2, 3, any of these three would produce these children. This would not produce those children. So we don't know the parent's genotype, right? Unless they finally do have a child with the recessive trait. And then we'll kind of get an idea. All right, let's move on here. There's more to this one. All right, scenario number 2. So not the same parents. We're going to look at different people here. We're going to go like this. Okay, here's a mom, not blue eyes, a dad, not blue eyes, first child, not blue eyes, second child, blue eyes. Well, we know that the blue-eyed trait is homozygous recessive, so 2 little e's. Aha! You have to ask yourself, where did they come from? Well, split them in half. One came from mom and one came from dad. That means mom has got at least one recessive trait, and dad has at least one recessive trait. We've now shown that because the parents have the dominant trait, that the parents have at least one dominant. They have at least one capital E for eye color. And because they had a child with blue eyes, that means they also have at least one trait, one allele for the recessive trait. So they both must be heterozygous. So when you have two parents who have dominant traits, you can't tell their genetic makeup, their genotypes, unless they have a child that has a recessive trait like this. All right, what about this kid? Yeah, this kid could absolutely be theirs. So if the parents are both heterozygous like this, the Punnett square would look like this, right? We just did this one a minute ago. Here is child number 2, and then child number 1 is any of these 3 right here. Any of those 3 could be that child there. So the question here is, could these be their child? The answer is absolutely yes. Yes, they sure are. This is the kind of logic and thing I want you to spell out when you actually do the lab. Let's go to a third scenario that actually has happened. I happen to have a friend who teaches high school at the very high school he and I used to go to. He is there, and this happened to him. He was doing pretty much, he was doing the high school version of the lab we're going to do, sent a list of traits home with the students, and one girl who has brown eyes took her list of traits home and came back the next day and had it filled out, and something didn't fit right. And so my friend said, you know, these can't be the case. You're going to have to go back to your parents and re-interview them. Look at them again. I think you got their eye color wrong. So she comes back the next day and she goes, I got the eye color right. They told me I'm adopted because it was this situation right here. How terrible would that be to find out you're adopted thanks to a high school genetics lesson? So the lesson there is really adopted people should know they're adopted. That's not a good way to find out. One of my friends found out she was adopted. She didn't find out she was in her 20s, and it freaked her out so much that she was literally institutionalized in a mental facility for a year. It blew her mind to find that out, just blew her mind. So bad idea. Okay, so here are the parents, both blue eyes, which means they are homozygous recessive. They do not carry the trait for non-blue. The only trait they can give is the trait for blue eyes. And here is someone that they raised as their offspring who has brown eyes, which is not blue. This person is either homozygous dominant or heterozygous. They have at least one of the dominant traits. Didn't get it from mom, didn't get it from dad. Okay, so here's doing the Punnett square just to prove it to ourselves. So here's the parents, they have blue eyes, which definitely means they are homozygous recessive. So we put their traits here. Here's mom, here's dad. You do the work and you go, look at that. Their children could only have blue eyes. This girl here, not so much. Let's scroll up here and see what's going on. What are the genotypes? I listed them out already. Okay, mom has homozygous recessive. What reproductive cells can she make? Only blue eyes. What reproductive cells can dad make? Only blue eyes. But this offspring here has non-blue eyes. Okay, what did the offspring receive from the mother? Well, must have received blue eyes since that's all the mother can produce and pass on. What did the offspring receive from the father? Blue eyes, since that's all the father, type that wrong. That's all the father can produce and pass on. Possible explanations of this: either this dad is not the father, this still could be the mother, right? That still could be the mother. So either the dad is not the father of the child, or the child's completely adopted. Those are the only two explanations for what's going on here. All right. So blue-eyed people can't have a brown-eyed kid, right? Nope. So either there was an adoption or mom's got some splaining to do. All right. So that takes us into, I'm still here, into doing some genetics problems. No, it doesn't. No, we got the fun part of the lab. We can't, here I am trying to jump ahead. What am I doing? Let me go get the lab here. What I did, because this lab is way too detailed, what I did here is I rewrote what I want you to do. The lab requires us to be in a room together and for us to figure out the gametes, or I should say the alleles on the gametes by classmates, and do a mating on paper, all right, to show what happens if you were to have a child with two of your classmates, right, two different children. We can't do that the way we're separate here. So we're going to do it a little differently. So let me move this here. So remember we did that, I'm still here, we did that hand folding thing. All right, I'm not going to read this to you, but you need to go to page 84 of the lab manual to read through this. The lab does not go over hand folding. I added that in here and I did an example just to show you. This is table 7.1, but I retyped it and it's there in Canvas for you. Okay, so if you were to, right, fold your hands and you found that your left thumb was on top, right, we're going to say you're left thumbed, you would actually have the dominant trait, right? Left thumb on top is either homozygous dominant or heterozygous, and we say you are left thumb, so that's your phenotype. Your possible genotypes, which I literally put right here for you, your possible genotypes would be either homozygous dominant or heterozygous, and then you rewrite the genotype. Now here's the rule, because we can't tell if you're heterozygous or homozygous dominant, we are, for this lab, we're going to assume that anytime somebody is expressing the dominant trait, we're going to assume that they are heterozygous. We're going to assume it, okay? It's written right in the rules, and I wrote it up here too. We're assuming they're heterozygous. So if someone's heterozygous, I recopied it here and put a colon, then what are the gametes they can produce? What do their sperm or egg cells, depends on what they produce, going to have? Well, a heterozygous person is going to make 50% of their reproductive cells are going to carry the dominant trait, and the other 50% are going to carry the recessive trait. This is how we're going to fill out this table. This is your assignment. Okay, so we need to look at these interesting things here, and this is where the lab falls down. They didn't illustrate any of these, but I did. So hang on while I go get that file and bring it up on screen for you. I need to change my camera. Too much paper on my desk. Let us go to, I'm going to go back to here. There I am. Okay, I'm going to pause the recording. Okay, we're back. So I have that file ready for you. I'm going to share that out. Only if Zoom will cooperate. There we go. Okay, so what this is going to do is explain some of those traits, well, all those traits that we have in the lab. I'm just going to go one by one here, and hair type, we'll come back to that one. Let's just skip ahead a little bit. Here we have something called widow's peak. Now, widow's peak is where the hair forms a funny little point here. So in the drawing on the left, widow's peak is either a homozygous dominant or heterozygous condition. And it's really obvious when you have it, unless someone also has male pattern baldness, which means they lose those hairs. I think I had it when I was younger. And the classic example of this is Eddie Munster on The Munsters, a show from the 60s. Hopefully you've seen it. Hilarious show. Look at that big old widow's peak he's got. All right. And then no widow's peak, which is the recessive condition. It's got to be homozygous recessive. And Gal Gadot is a great example of that. She does not have a widow's peak. All right. So we're actually going to have you look at you to determine what you have and fill it out on this form. Get the form that I made, not the lab book. I'm not having you fill out the lab book and turn it in. You need to look at the lab book, but I'm only having you turn in the form that I made. I mean, a shorter version of it. And I gave you much more space to work on. So it looks like it's more pages, but it's way less work. Okay, let's go to the next one here. Here are eyebrows. We just did this with the Levy family, didn't we? Bushy eyebrows, homozygous dominant or heterozygous, fine eyebrows, homozygous recessive. There we go. It's not a male-female thing either. It's equally common in males and females. It's just that very often females that have bushy eyebrows will pluck them, and then you don't know if they had bushy eyebrows. All right, so it's time to be honest with ourselves. We fill out our genetic table. All righty, color of the eyebrows as it relates to your head hair. So if your eyebrows are darker than your head hair, then you are homozygous dominant, 2 capital H's. If your eyebrows are the same color as your head hair, then you are heterozygous. Or if your eyebrows are lighter than your head hair, then you are homozygous recessive. When I had head hair, it was red. I might still have a lot of red in my beard, but male pattern baldness, another trait, when I was about 40, it just went boop, and in a couple of months it was gone from the top of my head. I used to have long hair, actually. Okay, but my eyebrows have always been lighter colored. I have blonde eyebrows which don't match my red hair. So I am homozygous recessive for this trait. All right, we can also look at length of eyelashes. Now, some people naturally have very long eyelashes and other people have very short eyelashes. So you know which one of these you are, all right? Very long eyelashes are typically twice as long as people who have what are called short eyelashes. Now, there is another genetic trait. We're not looking at this one in this lab, but I'm just going to tell you what it is because I'll bet you've heard of it. Some people have 2 rows of eyelashes, top and bottom. Isn't that wild? And so they have very dense eyelashes, and we call that, it's got some special name that means 2 rows. I forgot what it is. It starts with a die. We're not looking at that trait. Cool, but we're not looking at that trait. So if your eyelashes are pretty long, and usually, what is that? I think the cutoff is something like 1/4 inch or longer, I think is considered long, and anything shorter is considered short. So if you have long eyelashes, that is a dominant condition. You either homozygous dominant or heterozygous. If you have short eyelashes, you are homozygous recessive. Eye color, all right, we already went over that one. Eye color, you either have blue eyes or you don't. I have blue eyes. So other people have other colors. Doesn't matter what the color is. And as I mentioned before, so many students go, but what about green? No, green is not blue. If it were, it would be spelled the same way. Okay, so you either have blue or not blue. What color you actually have that's not blue is a different genetic trait we're not looking at right now. Just going to look at blue or not. All right, and then freckles on cheeks. This is real important because the lab manual doesn't say where the freckles are. These are freckles on your cheeks, not forehead, not chin, back, arms, cheeks only. It's totally different. And most people actually have cheek freckles. So freckles, cheek freckles, I should say, are a dominant trait. You either homozygous dominant or you're heterozygous. Having no cheek freckles is a homozygous recessive trait. Earlobes. So if you are free or unattached like mine, then you have the dominant trait. It's either homozygous dominant or heterozygous. Remember, we can't tell by looking at you. But if your earlobe is attached, right, it goes downhill, it's attached to the side of your head, then you have the homozygous recessive trait. Okay. And then dimples, usually only seen when you smile. So you smile and you get these little dimples up here. And it used to be that mothers would teach their daughters when they're young for years to hold their fingers in the corner of their mouths to make dimples. It was hysterical. My mom's mother, my grandmother, had her do that most of her childhood. And it didn't work because it can't. You can't change genetics. You either have them or you don't. It's muscles. It's muscles. Some people don't have the muscles that pull that part of the skin back to make dimples. You don't have the muscles. You can't do it. So if you have dimples, you have the dominant condition, which is homozygous dominant or heterozygous. If you have no dimples, you're homozygous recessive. Tongue rolling. All right. This is always fun because I always ask everyone to stick their tongue out at your teacher. So it's if you can roll the sides up into a little tube. Probably can't drink a soda out of that, but whatever. If you can do that without helping it with your fingers, then you are a tongue roller and you have the dominant condition. Some people cannot roll their tongue. They cannot teach themselves to do it because they don't have the muscles necessary to do it. There is another trait that's controlled by a different gene. It's really fun. If you're a tongue folder, you can take your tongue straight out and fold the tip back on itself flat. I've only had a few students that have had that. Pretty amazing. It's a really, really rare trait. Very cool though. Okay, and then we have sex at birth, female or male, and that is controlled by your chromosomes that you have. So we look at your chromosomes, and I showed you this last last lab. We have here someone who's got 2 of the X chromosomes, and we designate her a female. And then here's somebody with 1 X and 1 Y. Remember, Y is that deformed chromosome. That makes this person XY or male. All right, so it's sex at birth. You need to know that. It's either XX or XY. We don't use the dominant and recessive traits here for these. These are totally different. It's a different situation. Okay, and then I just threw a couple more in here that are fun. Hair type that I think I already did, but I made a little funny sketch. Curly hair is a dominant trait. Straight hair is a recessive trait. And heterozygous, the intermediate, is a blending, and you have wavy hair. And the degree of blending and the degree of waviness is actually controlled by other genes that we're not going to get into. Here's hand folding. It's got a nice drawing of it. So you can refer back to the recording here, figure out which one you are there. It's not, we don't require that one for the lab. Okay, if you have hair on your mid digit, which means you take your finger and you do this number, it's not on the last digit where your nail is. Nope, it's the next one back. It's called your mid digit. Here's the basal, the tip, and the mid. So if you have hair on this, you have to look on every single finger. Of course, your thumb counts. And look at those and see if you have hair. One hair counts, even one hair. All right, so if you have those hairs, you have the dominant trait. If you don't, you have the recessive trait. Okay, and another one, index finger length. This one's always fun. So what you do is you put your hand flat on the desk. And I'll just hold mine up so you can see it. You can see how my index finger is a lot shorter than my ring finger, all right, my third finger. So you compare first versus third. Looking at the drawings on screen, the far left one here, if the index finger is shorter than the ring finger, and it's usually very obvious, you have the dominant trait, and I use capital I here, so it looks kind of weird in this font. But if those fingers are about the same length, which mine are not, my first finger is much shorter, then you have the recessive trait. All right. So it can be about the same length. The first finger can be about the same length as the third, or even the first can be longer than it. That's the recessive trait. And then I just have the picture of the Levy family here that we already went over, so I don't need to do that. Okay, I'm going to stop our share here, and we're going to go back to the DocCam and take a look at the table that we're all going to fill out. Come on, Zoom. Fire it up. There we go. Okay, so here it's coming up. Boy, that's annoying. Good old Zoom. Okay, so here we go. Hair type. So if your hair is curly, wavy, or straight, you just use the word. These are all the phenotypes. You just write it in there. I'm going to do mine. My hair is wavy. Wavy. So what's my possible genotype? What's written right there? It's heterozygous. All right. What are the gametes that I produce? Or if you're the same way, well, first I'm going to repeat my genotype, ask you right there. And then I'm going to go, well, 50% of my reproductive cells carry the trait for curly, and the other 50% carry the trait for straight hair. All right. So I pass those traits. I have reproductive cells that have those traits. All right. I'm writing big here so you can see it. Normally you'd write a little smaller. Okay, widow's peak. Widow's peak, when I was young, I had a widow's peak. So I'm going to write peak here. And my genotypes are going to be, well, I'm either that or I'm that. Remember, for this lab, we're assuming heterozygous. So I'm going to rewrite that one and go 50% dominant trait, 50% recessive trait. All right. Eyebrows, bushy or fine, mine are fine. I do not have Eugene Levy style eyebrows, so I'm going to go fine. And I'm going to go, genotypes are these. I'm going to go 50% that one, 50% that one, or I could just, for this, I could just say 100% lowercase. Color of eyebrows, darker, same, or lighter than your head hair. Mine are blonde and I have red hair. Blonde is lighter colored than red, so I'm going to say that I have lighter. And I'm going to go with the homozygous recessive here, and then I'll rewrite it here. And I will go, I'm going to be lazy, I'm going to go 100% lowercase h. All right, eyelashes, long or short. I have short eyelashes, all right, which we all know of as average. That's probably a better way to say it, but the geneticists like the word short. Okay, so that means that I have the recessive condition. I'm homozygous recessive for that. I'll rewrite this here. So 100% little l. Eye color, I have blue eyes. So I'm going to go blue here. I'm going to write two little e's here. I'm going to rewrite them here and go 100% little e for blue. Cheek freckles, I am a redhead from Ireland and I am covered in freckles. So I'm going to say present because they are literally everywhere. I think I even have them under my feet. It's amazing. So I'm going to say present, and I will write down that I'm either homozygous dominant or I am heterozygous. For the lab, we're assuming heterozygous. So we're going to go that one. We're going to go 50%, capital F, 50% lowercase f. Earlobes, I have free earlobes. So my genotype is one of these. It's either homozygous dominant or heterozygous. For the lab, we're assuming heterozygous. Rewrite that here and put down 50% capital U, 50% lowercase u. Dimples, I have dimples. They're present. So my possible genotypes are either homozygous dominant or heterozygous. For the lab, we assume heterozygous. We're going to go like this, 50%, capital D, 50% lowercase d. Sorry, I'm writing this fast because I don't want this to go on for 5 hours. Tongue rolling, I can roll, so I'm a roller. I am either homozygous dominant or I am heterozygous. For the lab, we're assuming heterozygous. So 50% of my reproductive cells carry the trait for rolling, and 50% carry the trait for not rolling. I am a male, so I'm going to write that down here. I'm going to write XY. Okay, that means that 50% of my reproductive cells carry the trait for being female, and the other 50% carry the trait for being male. Guess what? All right, if you are female, then your genotype is written right there, XX or double X, here we go. And that means that 50% of your reproductive cells carry the trait for being female, but so does the other 50%. So we could actually just combine that and just go 100% X. So you females, you can only pass on the trait for being female. The sex of an offspring is determined by the father because he has one of each kind of chromosome. You ladies only have one kind of chromosome. You can only pass on the trait for being female, not the trait for being male. It's whatever the male contributes that determines the sex of the offspring. Okay, look at that. I got it done. Hey, guess what's up here? Your name. Okay, every page says put your name up here. Getting tired of reminding people, put your name on your pages before you submit them and your life will be better and I won't have to dock you some points. Okay, I got that done. Look at that. Don't lose this thing though. We need this. Also, you need to turn it in. Okay, now what we're going to do, because we're actually going to do a mating on paper, of course, and we're going to do like this. Well, normally we would use the traits of your lab mates, and there's really no easy way to do it. So I've come up with a different way to do it. First off, I'm going to put my name on there. First off, we're going to do here. So what I did is I've come up with the genetic traits for 4 of my favorite characters from Batman the Animated Series. You're welcome. This is Poison Ivy, my favorite character. Guess what? She's a botanist, and so am I, plant biologist. Okay, so possible parents here. We're going to use Pamela as a possible parent. So we're going to use your traits as one parent and one of the 4 people I chose as the other parent because we don't have access to each other's information. Normally, this is so much fun to do live in class. Okay, so Pam has, say, fill this out for them. I did all that research for you guys. See what I go through for you guys? So she has wavy hair. Her genotype, remember, we're assuming heterozygous if we don't know, right? But we know for hair waviness that heterozygous is the way. So she makes 50% of her reproductive cells carry the trait for curly hair, the other for straight hair, excuse me. Widow's peak, she does not have. So I did this. Anyway, I filled this all out for her, same as we just did. So there's Pam. Move this along. You have 2 females and 2 males, and I'll explain to you why I'm doing this here in a minute. I chose Harleen Quinzel. You know, her secret name is Harley Quinn. All right, I filled it out for her traits. And then we have Batman. Don't tell anybody, but he's really Bruce Wayne. Okay, so I filled it out for his traits. All right, and then we have the Joker. All right, if you're a comics person, you know that no one knows his real name because he never gives the same name twice. All right, so we don't really know who he is. Anyway, here are all his traits. So now we've got all his stuff. So we have enough to make a paper offspring. So here we have my traits, and we have traits for 4 different characters. And I'll do that over here, and I'll flip that back over. All righty. And so we're going to need that here in just a minute. The exercise that's coming up is called, very cleverly, Create a Kid. It's called Create a Kid. Okay. So what we're going to do is going to take the place of the Create a Kid procedure in the lab book from page 85 to 88. So use this instead. It gives a better explanation. I didn't spend a lot of time, but here's what we're going to do. So if you are female, your reproductive cells are female, all right, then you would fill out your information right here. What you would do is you go back to this first page, okay? You go back to this first page and you'd have a look at this column, right? This is the first page that we did together. You go and look at these results here, but we're not done yet. And you would put them here. That's if you're female. If you make female reproductive cells, you put them here. However, if you make male reproductive cells, you would put them here. All right. And if you don't want to put yourself in either of these columns, totally cool. No problem with that. Choose two of the people that I wrote up for you. So you can choose Poison Ivy hooking up with Bruce Wayne, or the Joker hooking up with Harley, or the Joker hooking up with Pam, that's her real name, or Batman hooking up with Harley Quinn. So you can use two of these characters. You need one male and one female, and then we're going to do it twice. All right. So you can use their traits. So I just care that you do the exercise and you learn something. And I know some people are not comfortable with considering themselves parents, all right, on here, and don't want to reveal some information. All right. That's okay. It's really okay. Okay. So, but we need to put just one thing here. So let me look at myself here under hair type. Here's, move that up. Here's hair type right here. All right. So 50% of my reproductive cells has the trait for curly hair and the other 50% for straight hair. All right. So I've got these. So which is it going to be when I produce reproductive cells, which is actually going to fertilize an ovule and pass on one of these traits? You have to choose one of these. All right. You have to choose one of these. And I forgot to bring a coin in here. Time to pause. Come on, Zoom, where are you? There we go. Okay, I'm back. I had to go find a coin. Geez, I don't have coins anymore. Okay, so we need a coin here. We're just going to flip a coin. I'm not going to do it on the screen here because it'll go flying through my room here. Maybe I'll try a short one. Let's see. Fingers crossed this works. So I'm going to flip this to see what's going on. And let's say that I flipped it and it turned up tails. All right. So here's the deal we're going to do. If it turns up heads, we're going to choose the dominant allele, and we would write that here. I'd write a capital C here. However, it turned up tails. Tails is going to be the recessive allele. So I'm going to write little c here and make sure you can tell them apart later. Then I'm going to do the same thing for my character here. I'm going to choose Poison Ivy because I like her. I'm going to go Poison Ivy. What trait does she have for hair type right here? I'm going to look across. Oh, look at this. She's the same as me. She's heterozygous. So I need to flip a coin here and choose one of these. So I get my coin ready, and I do it. Oh, and I got heads. You should do a better coin flip. I'm doing it small on purpose so I don't lose the coin. So, or hit the camera. So now I go like this here, and she came up as a dominant allele. All right. So this is what her ovule carries. This is what my sperm cell carries. They get together and they form an offspring that's going to be this plus this. It's going to look like that. And that means the offspring is heterozygous. And now I go, what's the phenotype of that? Oh, I don't remember. It was a long time ago, like 5 minutes. What's the phenotype of heterozygous? Let's see, heterozygous. Wavy, there it is right there. So now I write wavy. There we go. I just finished that one. See how easy that is? Super cool. Okay, now I go through, just in case you didn't get it, we'll do one more. Widow's peak, I'm gonna look at me. Widow's peak, I had a peak before I lost my hair, but there it is right there. So peak, because it's way over there, because I'm heterozygous for widow's peak, I'm dominant for widow's peak. I'm going to choose a heterozygous one. Remember, that's what we're doing. So heterozygous. So all the things you do here, you take from this column. So I'm going to have to flip a coin to figure out what goes in there. So figure out where my coin went. I'll do a coin flip. I got tails. That means it's the recessive condition. So for widow's peak, I'm going to write a little w here. And now I look back on poison ivy, and she doesn't have a widow's peak. She is homozygous recessive. She's going to make little w, so I know I don't need a coin flip. I make a little w for her. There we go. Little w and little w equals 2 little w's. There we go. This is homozygous recessive. What is the phenotype? We look on the page in case we forgot. It is absent widow's peak. So I'm just going to write absent here. Look at that. See how easy that is? So this is going to be the offspring. You guys go ahead and fill the rest of this out. And then that's one. Then we try to find our other page. There it is. Okay. And then we find the next page. Oh, there's that important name on there right there. Okay, now we do the second one. This is create a kid number 2. So you have to do it again. So you still have your information on hand. And now we want you to choose a different potential mate. All right, because we're not in class, we can't go around the class doing this, which really makes it hysterical when we do this. All right, so I'm going to choose Harley Quinn, and I'll do hair type. Okay, so hair type for me is, right, there's hair type right there. So hair type is, heterozygous. So 50-50, I have to flip a coin. So I get my coin out here. I'm going to do a little flip. Ooh, bigger flip. Oh, I got tails. That means it's a recessive trait. I put in this one here. I put in the lowercase w. I go look at Harley. And I go, all right, hair type. Oh, I did the wrong one. I did widow's peak, didn't I? I skipped ahead to that one. Okay, well, fine. I'll do that. Then we'll look at widow's peak. So her widow's peak is, let's see, she has a widow's peak. And so we need to choose the heterozygous condition. We need to flip a coin for her. Flip a coin, it turns out tails, which means she is also recessive. Little w, child therefore is a 2 little w's, right? Homozygous recessive. Their phenotype is absent. No widow's peak. All right. Let's do hair type because I meant to do that one first. Let's go back. We'll just do one more and then we can move on here. Okay. Hair type for me is wavy, which is heterozygous. So I'm going to flip a coin. Flip the coin. I got tails. And that means I put in a lowercase c for mine. Now we'll do it correctly this time. We look for hers and she is wavy, heterozygous. We need to flip a coin for her. I can just do it here. Up tails. So she is, her reproductive cell is here carrying the lowercase c. Little c gives us 2 little c's. Child's phenotype with regard to hair type is straight hair. Okay, even though I have wavy hair, she has wavy hair, had a child that has straight hair. Totally possible. Okay, continue that down. And then once you get to the bottom here, here's where it gets really silly. We want you to draw the face of your 2 children. I want you to name them. So put their names down here. Here's Ted and here's Alice. All right. Well, here's the fun thing that when you get down to the sex, let's do that too. Let's go and do that one. I should have done that one too. Let's fill that out. So father's allele, I am, right, XY, that's 2 different things. So I have to flip a coin to figure out what goes here. I'll find my coin. Oh my gosh, did I lose the thing already? There it is. Okay, because 50% of my reproductive cells carry the trait for male and the other 50% carry the trait for female. So I have to flip a coin. In heads, okay. So heads, and you can just randomly assign which one that's going to be. I'm just going to say heads is the first one that's written down. So let's go back to this. We'll say heads is the X. So we'll do that. So I'm going to say that my reproductive cell has the trait for being female. What does hers have? Let's go back to hers. Here we go. And she, being female, only makes gametes that are the X. So I have to just put X here. There's no flipping. So that means X plus X equals double X, which is female. Okay. That's good. Look at that. My second one is female. All right. And I called her Alice. All right. So fill this whole thing out before you make the drawing because it's silly to do it that way. I'm just trying to shortcut it to get this done and not have this be a 5-hour recording. Okay, now you go through, you look at the traits and you make a sketch of what they will look like and have fun with it. Man, over at Orange Coast College, we do the same lab. And I had a student who is an art major, and wow, is she good. She made the most beautiful sketches of her 2 potential children. I could not believe these things. And she just whipped them off during class. I think I told you before, I can draw insects and plants. I cannot draw people. She can really draw people. So anyway, you're going to want to go through with colored pencils if you have them. It's way better than doing this. You would look to see what color eyes they have. So you draw their eyes in and color the iris in whatever color. Eyelashes, you draw in whatever length of eyelashes that they have according to your characters that you drew. You draw in the ears. So if they have free earlobes, you draw that in. If they have attached earlobes, you draw those in. All right. So go through and do all that good stuff. We didn't do anything on nose, so nose is all up to you. If they have dimples, you're going to want to dimple those out. If they have cheek freckles, right, give them some cheek freckles, give them a funny nose there. If they have a widow's peak, well, incorporate that in the hair, because remember, our hair is either going to be curly, wavy, or straight. So you look to see what they have here. This one here, this is my second kid, has straight hair. So I'm going to go through and draw straight hair for this person. And again, I'm doing this super fast. There we go. So straight hair on the second one, and no widow's peak on the second one. So their hairline is going to be kind of rounded or pretty much straight across. It can be slightly rounded here, but not to a point. Okay, there we go. So I would do that hopefully with colored pencils. And that makes your 2 illustrations. So have fun with it. This part's really fun to do. All right, on to page. Wow. We might get this done before midnight. How nice. All righty. And that is that part of it. Okay, now we are off to a few questions here. Which parent determines the sex of a child? You should know that by now. You should know that by now. I have to say, my mom did not know. That's right. Did not know. Probably still doesn't know. It was amazing. And that would be the father. All right, the male determines the sex of the child. Females can only pass on the trait for being female. Males can pass on the trait for being female or the trait for being male. All right, so parent that determines sex of a child is always the male. Why doesn't the other parent determine the sex of a child? Well, in humans and other primates, the females don't even have the trait for being male, therefore they can't pass it on. So you'd say something like that here. And then you look at your children and say, what similarities do they have? So anything that they both have, kind of give it a quick note here. I don't need a paragraph, just briefly write it out here. Do your children look more like one parent than the other? I think that's kind of a fun question. All right, so look at them and see, wow, one looks like the Joker, stuff like that. That would be great. Moving on from that page. All right, now we're off to some genetics problems. So there are a million of them in the lab book. All right, so I only want you to do the ones that are on this document that I made for you, but we're going to do a couple out of the lab book so that you know how to do them. So I'm referring back to my notes here on the best way to do some stuff. There are multiple different ways to do genetics problems. Okay, so let's move this aside for now. I'm going to grab some more white paper. Maybe. Where did it go? There we go. Let me grab a little bit of blank paper here, and let's take a look at some of these problems that are in the lab book. I'm not requiring you to turn in the lab book problems. I made my own, but I'm going to use the ones in the lab book to teach you how to do this. Okay, so here's the actual lab, which again, you're not turning into me. Here's the table. I redid the table. You don't need to turn in that one. Let me move over here. Let's create a kid. There we go. There's the original. Okay, moving on. So you're going to skip all those pages because mine are better. There we go. So let me put this aside. And here we go. Here's practice genetic problems. So we're going to do some of these. I took these questions and put it on mine, but I skipped a couple that I didn't think were good. Well, actually, they deal with the lab we couldn't do remotely. Okay, Punnett square. I already showed you how that thing works, so I can just skip this page. Yay. Let's move on here. Let's try a few practice problems. Okay, so a green pea plant. Now, peas, I'm sure you know what peas are. They have beautiful flowers, and the flower color is a real interesting thing that is genetically determined. Let's see if this thing will focus better. Hang on one sec here. Sometimes it does, sometimes it doesn't. That's better. Okay, so pea plants have some really beautiful flowers, and this is really worded honestly bad. The whole plant is green, the whole plant is yellow. I don't get it. Normally we say they're flowers. Okay, so it should be that, but I don't know. We're just going to go with what's here, where they're seeds. You know what? That's what they mean. They're seeds. Because pea plants can either, the same species can have seeds that are either yellow or green. And it's interesting that when you buy peas from the store, like you might buy frozen ones or fresh ones or from the produce section, and you pop open that pod, the only ones they actually sell are green because people expect to see green peas, where yellow peas are just as good, but people don't expect to see them, so they freak out. So they don't sell the yellow ones. They only sell the green ones. All right, so this is going to be the actual seed, you know, a nice round seed, which can be either green or yellow. There we go. So what this is saying is that a pea plant that makes green seeds is this genotype. Wow, it's heterozygous. Well, that tells me right there, if this is green, that tells me that that is green and this is not. Let's keep going. Yellow plant is homozygous recessive. Aha. So two little g's. That means little g is not green, or better yet, it's yellow. Okay. So now we know the letters that are used. So we made them together, and they want us to do a Punnett square. Now remember, make this real easy. If it doesn't say which is male and female, you just figure it out yourself. You can just make it up. Doesn't matter. I'm going to write a nice table in here. I'm going to say a green pea plant. I'm going to put that one on top here, there. So remember, I split these two, capital G, little g. Here's the yellow one. We're going to do little g, little g. Okay, what if these two gametes get together? Then the offspring is going to be heterozygous. What if these two gametes get together? The offspring is going to be homozygous recessive. If these two get together, the offspring is going to be heterozygous. If these two get together, the offspring is going to be homozygous recessive. Okay, now they say, what's the genotypic ratio? And that means out of the 4 offspring type, that does not mean offspring, it means 4 types of offsprings these two parents could have, 1, 2, 3, 4. What are the ratios? Well, the genotype, first off, means you need to write down the genotype. So I'm going to do that right now. I'm going to write down heterozygous. I have, look at that, I have 2 of those. 1, 2 that are heterozygous. And then what's the other? Oh, the only other kind is homozygous recessive. And there's 2 of those. Okay, how many are there in total? 1, 2, 3, 4. So the ratio is going to be 2 out of 4 and 2 out of 4 there. You can simplify that down and say 1/2 or 50%. 50%. All right. And that's the answer to A. Phenotypic ratio means, what does it actually look like? Remember, what's its face? So this right here, what does that look like? That has green seeds. So we're going to go green seeds, 50%. And then we go, what's the other one? The other one's yellow seeds. Yellow. Oops, I'm writing the wrong place, aren't I? Green. Green. Oh no, my pen. Green. Oh my gosh. Oh my, nothing like a pen running out of ink right in the middle of this. Okay, I should have written that down here. So it's really green 50% and yellow 50%. All right, so genotypic ratio are the letters, and then phenotypic ratio, what does it actually look like? Okay. All right, a tall plant Homozygous dominant is crossed with a, oh, that's fun. It's crossed with a heterozygous tall plant. See that? They want us to do a Punnett square. I wish they would have left us more room. On the document I did, I made so much more room for you guys. Okay, we're going to take our tall plant. We're going to put it here. We're going to take the heterozygous tall plant, put it there. So what if this gets together with this one? We have homozygous dominant. This one with this kind, homozygous dominant. This one with this one, heterozygous. This one with this one, heterozygous. Okay, genotypic ratio. We just write down those letters. I'm going to let you write it over here. It's just easier. There we go. Genotype there is homozygous dominant. How many of those are that? 1, 2 of them out of 4. What's another genotype? It is heterozygous. How many of those? 2 out of 4 or 50%. Now we do the phenotypic ratio. And that is, oh, you're going to love this. They're all tall, aren't they? They all have the tall gene. So that means 100% tall plants. Love it. That's pretty easy. Okay, let's do another one. A white flower, homozygous recessive, is crossed with another white flower that's also homozygous recessive. So make a little Punnett square here. We're going to put those R's in. There they are. What if these two gametes get together? These two, these two, these two. Oh, look at that. Genotypic ratio, that's pretty easy. It's going to be 100% homozygous recessive. Phenotypic ratio is white flower, 100%. Okay. This is also known as true breeding when there's only one trait in the bloodline. So this would be called a true breeding white flower. As long as you cross 2 white flowers together, it'll always end up in offspring that have white flowers. All right. The same is true for some plants that have red flowers, and they're true breeding as well, where they don't have any other traits in the bloodline of those individuals. The ultimate example of this is dog breeding and cat breeding, where they are also, they're said to be pedigreed because they are true breeding. They are purebred. That's the term they like to use in the circles of dog breeding and cat breeding. They are purebreds. So purebreds are just from one line within the species. There are other ones in the species too, but those ones are true breeding because, or purebred, same thing, because they don't have any other traits. So we could actually say this: a purebred white flower is crossed with another purebred white flower, and you'd know what to do. Okay, same deal. A black chicken that's heterozygous is crossed with another black chicken that's heterozygous. I think the more important question is, why did it cross the road? Okay, I'll be here. I'll still be here. Okay, so we have our table. We're going to go heterozygous, heterozygous. What if these get together? Homozygous dominant, these, heterozygous, these, heterozygous, these, homozygous recessive. What's our genotypic ratio? Well, we have to write, I'm going to write it up here. How many of them are homozygous dominant? We have 1. And then how many are heterozygous? 1, 2. How many are homozygous recessive? 1. All right. What we have in this case, when we cross 2 heterozygotes, we get this interesting condition where the resulting genotypic ratio is 1 homozygous dominant to 2 heterozygous to 1 homozygous recessive. It's classic 1 to 2 to 1 ratio, classic thing. So that's actually a nice shortcut to remember. If you have 2 heterozygotes crossing, it's always going to be a 1 to 2 to 1 ratio. And then our phenotypic ratio is, well, what do we have here? These are black, black, and hey, that's a not black chicken. We'll just say it's a white chicken, or we can just say it's a black, non-black chicken, whatever we want to say. So we can go black chicken would be, how many of these are black? 1, 2, 3 out of 4. And we just, because we don't know the actual color. If we don't know the actual color, we just say non that one. So we're going to go non-black chicken. 1 out of 4 or 25%. Okay. You don't just assume what color it is if it's not stated. Genetics problems are very tricky. You've really got to read what they're asking. And right now I'm just giving you things that are really simple. See how they're getting a little harder? Homozygous dominant brown mouse is crossed with a heterozygous brown mouse, and tan is recessive. Look at that. They're letting you figure out what letters to use. Okay, that's good. I'm going to say that's, I'm going to use M for mousy. Okay, so homozygous dominant brown mouse. I'm going to say our mouse is 2 capital M's, and the heterozygous brown mouse is going to look like that. Right, heterozygous brown. They're both brown. Homozygous, yeah, got it. Okay, now do our Punnett square. We can write that out. I'm going to put that one up here because we don't know the gender. And again, I like sex. Sorry, I really like to put the female here and the male over here. Just a standard I developed when I was doing these in college a million years ago. There we go. Okay, set it up. We have the breeding. We've got homozygous dominant, heterozygous, heterozygous. Genotypic ratios. Well, I'm going to write down that homozygous dominant and go, there's just 2 out of 4. Any different things here? Yeah, we've got heterozygous, 2 out of 4, and you can resolve that and go 50%. There we go. Those are our genotypic ratios. Our phenotypic ratios are brown. They're all brown, 100% brown. Now, this page, even though I'm not asking you to turn this in, I'm just trusting you to do it because it's super good practice. It really trains your brain. You really get into it. Let me just move that over. I am not going to do the rest of them for you. You should, just you get into it. Now, this next section, unfortunately, they use the wrong term here. It's not really called dihybrids. Di is 2. This is supposed to be called 2-trait crosses. A cross is a mating. I don't know why we use crosses instead of 2-trait mates, but this is what we do. Anyway, that should say 2-trait and not dihybrid. Dihybrid something else. Okay, so what it's saying here is that what if you look at something more advanced that has, oh my gosh, 2 traits at the same time? Whoa, yep, we can do it. Got to stay with me though. This can be a little complicated, so stay with. All right. Here's what we're going to do. So he has steps here too. Now, he's using another way to figure this one out. I'm not going to turn the page yet till I finish what I'm saying. I'm not a fan of the fraction method. I'm not a fan of it. And you'll see it in just a sec when I move this page. I'm not a fan. I'm not even going to teach you how to do it because I really don't think it's the best way to do it. It confuses me. I am not a mathematical genius. And so honestly, I don't like the method that he used. So we are going to do this stuff here, but we're not going to do this fraction thing. If you already know how to do it or you like doing that method, you are welcome to do it, but I am not going to push it. So let me move this over. Here we go. So this is page 92. Remember, I want you to refer to the lab manual when studying all this, but you only have to turn in the document that I gave you filled out. Okay, this is the method he's talking about. And I had to really stare at this for a while to figure out what in the world's going on. This cross tree method, no, thank you. You can do it if you want, but I just don't get it. Okay, it doesn't make sense to me. So we're not going to use that. I am not going to use that. Again, you're welcome to if you want. And then he's got some problems. Here's what we're going to do. I am going to bring in my own notes here and show you 2 other ways of doing this that I think both are better. All right, it's just me. Okay, so here are my notes. I've got this here. All right, now this is going to get a little weird. If you've never done 2-trait crosses before, you probably have in high school because you're supposed to have had this stuff in high school too, but you may not remember it. Okay, so here's what we're going to do. We're going to look at some seeds of pea plants, and we're going to go seeds of pea plants. Why do we use pea plants so much? Because the original person that studied and figured out all of genetics used pea plants, and we've been using them ever since. They're beautiful plants. They're easy to grow, and they're really easy to see the traits, and it works out. All the math works out. Okay, they can either have round seeds or non-round seeds. I know it sounds funny, but round, or they can be oval. They're never square. That would be hilarious. Okay, so round seeds are indicated by a capital R. So that can either be homozygous dominant or heterozygous. Non-round is always going to be homozygous recessive. Okay. And now what about the color of these seeds? The color is going to be either yellow, homozygous dominant, or heterozygous. And the non-yellow, I mean, frankly, they're green, but we're just going to go with non-yellow. That's what he wrote in the lab manual. And they are always homozygous recessive. Okay, there's what we're going to do. We're going to do something with some seeds. That's our example I'm going to show you. So what if we have two plants that produce seeds that are like this? They do. Okay, so this is one plant, and it is, let me pull that back down a little bit. It is heterozygous. All right, here's our plant. Another bar across here so we can really see the difference. Like, here's my intro. So it's heterozygous. It has round seeds, and it has yellow colored seeds. So the seeds are round and they're yellow and they're heterozygous. All right, they're right here. So he, in his example, he gave us this genotype. We didn't have to figure it out, but you should be able to based on the key up here. All right, and he bred two plants that have the same kinds of traits together. So we did this. We usually do an X or an asterisk or a dot or whatever to indicate multiplication or mating. And so they actually have the same genotypes of these two plants. All right, how do we do this with two traits? Well, there are two methods to figure out this first step. The first thing you have to do is form gametes. All right, which is true for anything that has sexual reproduction. The first they form gametes, then they have to find a mate, then they have to mate, and the gametes have to get together to produce an offspring. Okay, first step's always form gametes. So how do these form gametes? Well, a gamete can't have all of that in there. It's only going to have one R and one Y in every gamete. So there are two pretty straightforward methods for figuring this out. The first method, right? So method number one is to use a Punnett square. And sorry, my writing is kind of sloppy. I'm trying to go so fast so that we're not here till midnight. It's already, oh my gosh, it's already an hour and a half after my dinner time. So here's our Punnett square. We're going to do a Punnett square. So what you do is you first separate the traits. So here's what I do. I like to take a red pen or pencil and I make a big line right there. I separate those traits. So the R's are together, the Y's are together. Okay. And then I need another color. Here we go. Then what I do is grab another color and I say, okay, now I'm going to separate these. Okay. So I've got a big line, big red line separating these. That red does not come through very well, does it? Let's do a little bit. There we go. Okay, I'm going to separate them like that. There we go. And then I'm going to make a table. So I form a Punnett square and I'm going to go, all right, so this individual, I'm going to make its gametes, and then this individual, I'm going to make its gametes, and then later we put them all together. So I'm going to make a little Punnett square. It doesn't need to be real big. I'm going to take the R's and put them across the top. See what I did there? I separated them and I put them across the top. And then here, I'm going to take the Y's put them down here. And now when this individual makes gametes, one gamete is going to contain the capital R and a capital Y, and that is one cell. So that's either a sperm cell or an egg cell. And then another one's going to contain the little r and the big Y, and that too is either an egg cell or a sperm cell. And another one is going to have capital R and little y. That's either a sperm cell or an ovule cell. These two get together, it's going to be this, and that's going to be a sperm cell or an ovule cell. And these four are going to get used here in a minute. And now we do it again for the second plant. I know they're the same one, but I'm still going to write this out a second time because we need the practice. All right, so just bear with while I do this. There we go. Okay, there's our four reproductive cells. I know they're similar, so these are going to be the same thing. All right, and now we know what gametes are going to be formed. And the second way of figure out what gametes are going to be formed, okay, we'll use them in a minute, but first we're going to generate a method number 2. That's a really nice 2, isn't it? Number 2, geez. Number 2, there we go, is to use FOIL. Okay, yes, you people that are fans of good old algebra, you know what FOIL is. First, outside, inside, last. Yeah, baby. So I'm going to rewrite the genotypes down here. There's that one. And here's the other one. It's the same, but I'm still going to write it down here. Okay, so for our first plant, we're going to use FOIL. And here's how FOIL works. It's pretty sweet. And there's less writing, actually. So the first kind of gamete this makes is first, which is the first of these two pairs. You go this one and this one. Give us capital R, capital Y. Outside, this R and this little y, it's going to give us that. Inside, this little r and that big Y. And then first, outside, inside, and then last, we go this R and this Y. We go like this. There we go. And now we have the gametes, and it's exactly the same answer as that one. So this is two different ways to figure out the gametes, and FOIL is a lot less writing, actually. So then we do it for this one here. First, outside, inside, little r, big Y, and last, little r, little y. There we go. They're the same thing. Okay, so that's the thing that you can do the gametes in either way there. Gonna need some more paper here. So I'm gonna go over here. And now here is the messy part. You need some paper. All right, you're gonna need scratch paper for doing this. By the way, there's not enough room in the lab, I noticed. So now what we do is we put together the mother of all Punnett squares. Well, no, not really. It's not that bad. Okay, we're gonna take our information here. Well, how many gamete types are we going to do here? 1, 2, 3, 4. Great. So we need a 4x4 Punnett square. So we're going to go here, here. 1, 2, 3. That'll be 4. 1, 2, 3, 4. There we go. Now I'm going to write these on the top of it. All right, remember these are all the same. So I'm just going to take this one, either one of these, doesn't matter. They're all the same. I'll do it up here. I'm going to go R, capital Y, check, because I know I got it. Little r, big Y, check. Big R, little y, check. Little r, little y, check. And then we know that the other parent actually has the same thing. I'm going to write those down this way. Big R, big Y, check. Little r, big Y, check. Big R, little y, check. Little r, little y, check. I got them all. And there's our Punnett square. I know it looks like a monster, doesn't it? It's not that bad. It's not that bad. You just have to go follow the process. Don't be watching TV when you do this because you will definitely stumble. Okay, now you have to remember, always go back. This is a mating. This is an ovule. Remember, I like to put the females on the top, males over here. So stick a little tail on there. although these are plants and plants don't have tails on their sperm, but why not? Okay, so here we go. If this sperm cell gets together with that ovule cell, the offspring is going to be big R, big R, big Y, yay. This one's going to be, what if this one fertilizes that kind? It's going to be big R, big Y, big Y. How about this one and this one? Big R, big Y, little r, little y. How about this one? Big R, big Y, little y. And you just keep going. All right, this one here is going to be big R, two big Y's, little r, big Y, big R, big Y, little r. Over here, two little r's, two big Y's. These two, it's going to be two big R's, big Y, little y, a big R, a big Y, a little y. These two here, big R, little y's, big R, little r, two little y's. This one and this one, big R, big Y, little y. This one and this one, little r, big Y, little y. This one and this one, big R, little y's. This one and this one, everybody's little, and we just did it. We just did it. Okay, so to figure out our percentages, because you know I'm going to ask you that, you figure out how many offspring there might be. You look in here, you just count the boxes. It's super easy. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, blink. So our ratios are going to be anything out of 16. So it's some number that we count out of 16. All right. So let's move this aside here. Oh boy. All right. Now the next step is to take inventory of the genotypes. Inventory of the genotypes. This is going to take you a little time. So you just start writing stuff down. I've got that one there. So I'm going to write it down over here. I just need a little space. Just going to rewrite it. Or you could just, no, it's better to rewrite them over here. There we go. That's this one. And now check mark that when you put a tally in, right? Easy to lose your count. So you write it down, put a check mark, put a little 1 there, and now look to see are any more the exact same thing. Nope. So there's only one of those. All right. So 1 out of 16 is this genotype. That's the genotypic ratio. Now we'll look at the next one. Let's go to this one here. Let's go big R, little r, two big Y's. And I'm going to go check and give it a 1. And now I look to see, are there any others that are big R, little r, and two big Y's? Oh, there's one right there. Okay. Check, check. We have 2 of those. Let's see if there's any more. You look around. And there you go. Okay. So now we have 2 out of 16 are that genotype. Now we go to the next one. Let's go this one here. Let's go 2 big R's and a big Y and a little y. And we have 1 and we check it. Okay. So we have big R, big Y, big R, big Y. There, nope. Big R, big Y. There it is, right there. It's 2 of them. Big R. Okay, that's 2 out of 16 are that one. Did I check both of them? 1, 2, 3, 4, 5. Good. Okay, now our next one. Look at this guy here. Big R, big Y, little y. Okay, check it. Put a tab. Now we're going to go big R, little r. Okay, there's another one. Big R, big Y, little y is another one. Big R, little r. That one right there. 4. Big R, little. Yep, there we go. We have 4 out of 16. That's that one. Okay. And now we go to, what's the next one here? We'll take that one right there. I'm just going to make a little line. I'm going to do this. Okay, 2 little r's, and two big Y's, two little R's, two big Y's. There's another one that's two little R's and two big Y's. Okay, there's two of those, two little R's, two big Y's. One, sorry. Isn't there really? Hold on here. Two little R's. I think I miscounted. Two little R's and two big Y's. Two little R's. Oh, yeah. Two little R. Okay, I miscounted. That's 1 out of 16. Okay, there's that one. And now we go, here's another one here. That is R, two little Y's. We have one of those. Oops, sorry. Okay, so two big R's, two little Y's. Two big R's, two little Y's. Nope, just one. Right? Two big R's, two little Y's. Yep, that's 1 out of 16. Okay. And then who's next here? We have big R, and two little Y's. So big R, little r is our first indication right there, right? Yep, that's another one. It's two of them. And there's no more like that. So that's 2 out of 16. Okay, what's our next one to write down? Here's one right here we haven't done. Two little R's, a big Y, and a little y. I'll check that off. Two little R's, a big Y, and a little y. Two little R's, a big Y, and a little y. I'm going cross-eyed looking at this thing. Two little R's, a big Y, and a little y. Two little R's, big Y, there's their one. Two little R's. Speak out if you guys see it. Oh, okay, I think it's 1, so 1 out of 16. All right, I'm assuming I got them all. And then this one, two little R's, two little Y's. Check that, put a 1, two little R's, two little Y's. So everybody is recessive here. Two little R's, two little Y's. All right. And if you did this right, this should add up to 16. You should be able to go 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Ha ha, I missed 2 of them. Okay, well, trust me, they're there somewhere. Again, I'm trying to go really fast. So I miscounted somewhere. We're just going to move on. I'm going to trust that you're going to do that. So now what you have to do is figure out the phenotypic ratio. So what's the phenotypic ratio? So now you have to go back to the origin of this lesson here and look to see what do all these things mean. All right. So let's go here. So okay, Zoom cut me off right in the middle of a sentence. So I hope it's going to continue to record now. Okay, so what we have here is we have to translate all this stuff. Whoop, where are we at here? So we have to go back and translate this, not that one, and figure out what all these things look like. So this one right here, these R's, that's going to be round. So round, what color is that? It's going to be yellow. All right, I write yellow in there. And then I go here. This one is also round. It's heterozygous. So this is also round with yellow. So this is the same thing. This one's round with yellow, same thing. Round with yellow, same thing. Okay. So I'm going to rewrite that down here. Now that I know I've got a whole bunch. Round with yellow, I have all these ones. This one is going to be not round. So not round here, but it does have yellow. Okay, not round and yellow. So that's another one, not round and yellow. All right. This one is going to be round, not yellow. Round, not yellow. No, yeah, not yellow. Okay, that's one too. Round, not yellow. Okay, that, and this one's round, not yellow. This one is not round and yellow, right? Not round and yellow. Yeah, not round and no, this one's not round and not yellow. I screwed that up, didn't I? That's what happens when I go too fast. So this one is not round, and oops, plus it is yellow. This one is not round and also not yellow. Okay, now we have, okay, there's our possibles. How many are round yellow? 1, 2, 3, 4, 5, 6, 7, 8, 9. Well, it's if I got it right, 9. How many are not round and yellow? Not round and yellow. Not round and yellow is this one, not round and yellow. It's going to be 1. There's our 1 there. How many are round and not yellow? Round and not yellow. I think it's probably a couple of the ones that I wrote down improperly when I put this table together here. Just skip that for right now. How many are not round and they are yellow? Not round and yellow. So it's going to be, I think it's just this one. I think it's just one of those. And then how many are not round and not yellow? That's also going to be 1. So that's how you do the phenotypic ratios. You just look and see what they actually mean and write it down, like write it down here in a list and count how many here, how many actually have that trait. All right. Okay, that's how you do those multiples. And that's why, wow, they are a lot of work and you need some scratch paper. Okay, where are we? We are on, that is my 3rd page of notes. Here is my 4th page of notes. And I did the same thing on the 4th page of notes. Okay, that's how you do those guys. Let's go back to the lab manual itself. Here. All right, what is this one here? That's page 10. That's too far along. Let me find my pages here, everybody. Bear with me. You can't imagine the mound of paper that's on my desk right now getting through this. It is crazy. You probably have a similar mess on your desk. We're all in the same boat, everybody. Okay, here we go. Where is the rest of this? There's 9. Okay. No, I know what I want to do. I want to get the lab itself. I do want to do this. Okay, we're back to the lab. There we go. Okay. So again, this cross tree method, I'm not a fan. If you want to do it, you're welcome to, but I don't get it. Okay. There's a rabbit problem using the 2 trait cross. Here's where they want you to write out the phenotypes of these. Here's male rabbit and female rabbit. They've got all the info, not enough room to do this, really need scratch. Here is a cyclops. That's this weird looking thing. It lives in ponds. Very strange looking animal. Go through those. I'm not going to do those ones for you. Okay, and now we come to sex-linked, which is also called X-linked. There are some Y-linked things too, but we're just going to look at the X-linked. Here's how these things work. These are really interesting. First off, consider our chromosomes. So you've got the chromosome. Remember, the thing winds around like this. All right, you get our 2 halves. They kind of wind around, and that the gene is on part of the chromosome. All right. So with these things that are sex-linked, there are some traits that are only on the X chromosome and they're not on the Y chromosome. Remember, the Y chromosome kind of looks like that. Looks like the letter Y. It's a stumpy thing, and it's missing all kinds of parts. All right, they're gone. Well, that gene is here on the gone part. So that means that if something is a male, if it has that funky Y chromosome, it doesn't have that gene. It doesn't carry that gene. Its X chromosome does carry that gene. So there are lots of traits for which males have, and it's only because that trait is on the X chromosome that they got from their mom. One of those is male pattern baldness, losing one's hair right around the age of late 30s, early 40s usually. Now, that's right, male pattern baldness. I inherited that from my mom because my X chromosome came from my mom. My Y is from my dad. You cannot inherit male pattern baldness from your dad. Nope, you get it from your mom. You get it from your mom. All right. So it's carried on the X chromosome. These are sex-linked or sometimes called X-linked. Well, these little flies called vinegar flies, they are not fruit flies, they're vinegar flies. They have the same kind of condition for eye color. So in these flies, eye color is a sex-linked trait and red is dominant to white. So a normal fly has red eyes. Doesn't that sound kind of spooky? A fly with red eyes staring at you? Yeah, well, that's normal. Okay, but white is recessive. So the recessive condition, recessive equals white eyes. There we go. Okay, they set it up. So this is how we write this trait too, by the way. When something has a trait that's sex-linked, we write that trait as an exponent. See that right there? I'm going to recopy for you. This is that X right there. We write it there as a superscript, right? Exponent right there. So this one is big X, little r right there. So what they're asking you to do here, and you really should practice this, write out the sexes and eye colors with the following genotypes. I don't know why there's a question mark there. Okay, so here it is. Here's the trait. This is a female, 2 X's, and carrying the trait for red eyes and the trait for white eyes, but because it's heterozygous, this individual has red eyes. So this is a red-eyed female, and this is the symbol for female. So red-eyed female. This one is a male, XY. This is a red-eyed male. This is the symbol for male. Notice how the trait is only carried on the X chromosome. So you're just going to write X with an R up here. It's literally that R up on that arm. And then the Y, you don't put anything there because there's no trait on it. Okay, here is another female, and she has, look at that, both the homozygous recessive condition. She is a white-eyed female. This one is a red-eyed female. This one is a white-eyed male. Okay, it's a little R right there. That's what we want you to do right there. Okay, so now I want you to go backwards and write out what's a genotype of a white-eyed male. Well, if you did this, you know what to put here. Genotype of a white-eyed male is X little r y. A red-eyed female that's heterozygous. Okay, red female, there's one. Oh, she's heterozygous. There it is right there. Female, red-eyed, heterozygous. We got it. White-eyed female. Well, first off, female, 2 X's, and then it's going to be 2 little r's. Red-eyed male. So we got male, red-eyed male. It's an X, Y, and it's going to be an R up high on this one, a capital R, and we just did that part. Now it goes show the Punnett square. Okay, so same deal we've done before. This is a simple 1 trait cross. We're going to go female. Remember, I like to put the female up this way, and she is recessive. Red-eyed male is going to be XY. That X is going to have a capital R on it. And now we do the crosses. If the sperm cell carrying this fertilizes an ovule with this, it's going to be, I'm going to write the X is kind of small and the R is big so we can see them better. It's going to be heterozygous for the trait, and it's going to be female. This is going to be female, heterozygous for the trait. This is going to be male that is recessive for the trait. Remember, the Y doesn't carry it, so you don't write anything there. This is going to be another male, recessive for the trait. There we go. Okay, genotypic ratio. We write down what the genotypes are. We write that one, that one. We write this one, this one. That's all. Yeah, okay, there's only 2 different genotypes. There you go. So we have 2 out of 4 or 50% that one, 2 out of 4, 50% that one. And then phenotypic ratios. Okay, now we write it out. Excuse me. So what do we have here? This is going to be a female. Look at what we did already. We can just check it out. Female that's heterozygous like this. There we go. Red-eyed female. So we have red-eyed female, 50% percent. And then this one's going to be a white-eyed male. White-eyed male, 50%. Boom, done. Bam. Okay. And then it just goes on and on about that. And that's X-linked. Now, the things that are sex-linked are things like male pattern baldness. I think he's got more examples here. Hemophilia. That's why only males can get hemophilia. Females cannot get it. There's no such thing as a female hemophiliac. It's a male condition that is sex-linked. Muscular dystrophy is sex-linked. And some forms of color blindness are sex-linked. Usually red-green color blindness is sex-linked. Males get it, females don't. Okay, in fact, there's hemophilia right there. I'm not going to go through these examples. The calico cat example is a lot of fun because I think we had a calico cat. I love them. They're beautiful. All right, so you can go on down there and you can do that. You don't have to do that one. I'm going to keep going here. Incomplete dominance is a lot of fun. So incomplete dominance is not where things are completely dominating the other. It's where one trait, the dominant one, and the recessive one blend together to make something intermediate. And that is something that you might want to do. You might actually want to color yours, and that will really bring it home for you. This one is red, this is white, and in snapdragons, you breed those together and you get pink. You get pink colored snapdragons. There you go. Okay, they're a lot of fun. So how do we do this? It's the same kind of thing. It's just you interpret the heterozygous condition differently. So a red snapdragon, a red flowered snapdragon, is mated to a white flowered snapdragon plant. Show the Punnett square. All right. Okay. Did they give us the letters? Sure they did. They're right there. So here's our red. We're going to take, this is really saying this with white, this. So now we know how to do that one. We just take our letters here and here, heterozygous, 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 heterozygous. Wow, they're all heterozygous. The genotypic ratios are 100% heterozygous. Phenotypic, well, they're all pink, 100% pink flowers. How fun is that? See how easy that one was? This one is codominance. Now, codominance is another situation we haven't encountered yet. And the classic example for this is the ABO blood groups in humans. Now, you have to think of this in terms of blood cells. So let's think of red blood cells. Red blood cells are round. There we go. They're round. And there's going to be 4 different conditions here. 1, 2, 3, 4. Back in the early 30s, they discovered that some red blood cells had these funny little extra chemicals on them, and they kind of form a shape. So I'm going to draw them as a little square here. All right. And because they didn't name it something fancy, they said this extra thing is going to be chemical A. And so anyone's red blood cells that had chemical A on them, we say that they are blood type A. It means you have this extra chemical around your red blood cells, and they call it A. All right. And then not long after, they discovered some people have a different chemical with a different chemical shape. I'm going to illustrate that by these little triangles. And they said, oh, some people have this kind, and we're going to call that B. And then they found that some people had B. I colored in so you could see it better. They had B, and at the same time, they had A. They had both of them. That's one busy red blood cell, huh? And so these people were said to have AB blood. And then some people had neither A nor B. Neither A nor B. Neither A nor B. they have exactly 0 extra chemicals around their red blood cells. So we say that these people are blood type 0, but people freaked out. What do you mean I'm a 0? That's not very nice. And so they said, we meant O, your blood type O. Blood type O is not a kind of chemical. It's not a chemical on the red blood cell. It means you have no, you can read it as no if you want, no A or B, neither A nor B. They're just a plain old red blood cell. There you go. And this is how they write it. Remember, you get these different things from your parents. So if the mother has A or B or O, and the father is A or B or O, and these get together, because remember, you have 2 copies of everything. If someone has 2 A's, right here, they're homozygous dominant, but we don't use dominant term because they're all dominant in this except O. So if someone's got A and A, we say that they are blood type A. If they have A and O, they still have blood type A. If they have B and A, we say they are AB. We always put the A first. If they have 2 B's, they are blood type B. If they are BO, they're also blood type B. And here's a typo you need to fix in your lab manual. This one right here is wrong. The typo point right there, B and O makes B, doesn't make O. And then if someone's O and O, that's right. What, you got one from one parent, they're O, that's just blood type O. So the blood types are A, AB, B, and O, and that's what they are. And we can figure out how you got these based on your blood type and your parents and or your siblings. And we can figure out what your blood type is. You should have your blood typed, actually. Everyone should carry an identification card with them telling what their blood type is. It should definitely be on file with your doctor so you can always find out. Oh, I see a typo here. Rink dominance. Okay. And that's in here. So what he's got as an example here, it's kind of fun. He's saying if 2 individuals with blood type A, B marry, okay, and have a child. So here's A, B through Punnett square, A, B. What are the genotype and phenotype ratios of the children? Where we can have A, B, A, B, B, B. So the genotype ratios, are going to be AA is going to be 1 out of 4. AB, 2 out of 4, BB, 1 out of 4, okay? Phenotypic ratios would be the same thing, okay? Pretty much. We just say for the phenotype, we just shorten that to A, shorten that to B, but we always call that AB. AB is actually very rare blood type. Okay, this is a fun one here. Disputed paternity case. Ray's new baby has a blood type of O. Oh, okay. So we've got a father, which we normally use with a triangle, and a mother, circle, and they had a kid, a branch. New baby is blood type O, her blood type, so the baby is also an O. So the baby has O. No. Ray's new baby has a blood type O. Yeah, her blood type is B. Okay, I think they mean Ray is blood type B. And Kylo Ren's is A. There we go. Okay, this is Kylo. This is Ray. Okay. And then, so we're setting it up with, oh, I see, disputed parentage, huh? O, B, that's Ray. And this one is Finn. Okay. Well, this is fun. Okay. So here's the disputed parentage. They're wondering who's who. Kylo Ren is A, Finn is AB. Finn insists he's the child's father. Oh, could that happen? Could that be right? Guess what? This really happens. Okay, well, let's look at this. Ray, we know Ray is the mother of this kid, right? She gave birth to the kid. So Ray gave birth to a kid who's O, which is really O. That means Ray, what did Ray give to the kid? Ray gave the kid an O. So Ray must be BO. Kylo is A, but we don't know his second allele. Well, the kid got one O from Ray. The other O came from the other parent. Look at that. Kylo can be the parent of that kid. Let's look at Finn. We've already figured out that Ray must be BO and the kid has to be OO because O blood is always OO. Finn is AB. So if one O came from Ray, the O had to come from the other parent. Finn doesn't have O. So Finn is not the parent of this kid. All right, you could figure it out logically. It's pretty straightforward and lots of fun. Okay, let us move that aside. Hope the recorder's still going. You're listening to this. I'm not done yet. I'm almost done, so close. I'm just trying to find the last page of the docket. I can't find it. Desk is literally buried, guys. This is literally buried. I can't get to this stuff. 9, 10. Okay, here we go. All right, I think I found it. Okay, so here are all the ones right here, all the ones you're going to do and turn in for me and putting your name on the page, top of every page. Okay, so this is complete dominance. You're going to do this one here, put it all down there. Here's 2 trait crosses using rabbits. We just did an example of that. It's going to take you a little work. Here's that giant Punnett square you're going to do as part of the rabbit one. And going down, here's sex linked. I'm giving you a different, all these are different than what's in the book. Sometimes I use the same organism, but they're not the same problem. That's why you should practice. Turn that over. And then here are the sexes for the flies, because I use the flies again, because I thought they're pretty cool. Punnett square, bunch of questions about the flies, because they're a great example of sex linked, which also works for other stuff. Incomplete dominance, where I use carnation plants instead of snapdragons. All right. And then we go to the next one. Here's still the carnations. And then codominance, here is that last one with blood type, but it's a real live paternity suit. And you're just going to have to wait to see it to do it. I think you'll really enjoy it. I use it in all my classes. I think it's really fun. Okay, let me get my camera sorted here. There we go. I'm back. Okay, hopefully this has all worked and you're still with me and not nodded off. My voice is about done. Thank you very much for watching. Have a good evening. That's what I'm going to do. Bye-bye.